Chapter 5: Asking Clinical Questions. Research Methods. Medical research typically aims to discover the relationship between one or more events or characteristics, such as being exposed to a toxic substance, having a family history of a certain disease, or taking a certain drug, and others, such as contracting or overcoming an illness. All these events or characteristics are called variables. Independent variables are presumed to be the causes of changes in dependent variables, which are presumed to depend on the values of the independent variables. Research, therefore, typically seeks to uncover the relationship between independent and dependent variables. For example, in studying the effectiveness of a particular drug for a disease, the use or non use of the drug is the independent variable, and the resulting severity of the disease is the dependent variable. Unless a research project Unless a research project studies an entire population, it will involve the use of a sample, a subset of the population. Most samples used in biomedical research are probability samples, samples in which the researcher can specify the probability of any one element in the population being included. For example, if a sample of one playing card is picked at random from a pack of 52 cards, the probability that any one card will be included is 1 out of 52. Probability samples permit the use of inferential statistics, while non probability samples allow the use of only descriptive statistics. A sample is representative if it closely resembles the population from which it is drawn. All types of random samples tend to be representative, but they cannot guarantee representativeness. Non representative samples can cause serious problems. A classic example of a non representative sample was an opinion poll taken before the 1936 US presidential election. On the basis of a sample of more than 2 million people, it was predicted that Alfred Landon would achieve a landslide victory over Franklin Delano Roosevelt. But the result was the opposite. The problem? The sample was drawn from records of subscribers to a literary magazine and telephone and automobile ownership. And such people in that Depression year were not at all representative of the electorate as a whole. A sample or result demonstrates bias if it consistently errs in a particular direction. For example, in drawing a sample of 10 from a population consisting of 500 white people and 500 black people, a sampling method that consistently produces more than 5 white people would be biased. Biased samples are therefore unrepresentative, and a true randomization is proof against bias. This kind of bias is called selection bias or sampling bias. Bias from non-representativeness Bias from non-representativeness may be due to simple errors in drawing the sample, as in the 1936 opinion poll, or due to more subtle influences. Patients with a certain disease who are referred to a specialist or to a teaching hospital are likely to have more severe disease than other patients in the community with the same disease. This is referral bias. Patients without insurance may not be diagnosed or come to clinical attention at all. Self selection may also occur. Certain kinds of people may volunteer to take part in a study or be attracted by payment for participating or by the promise of free care, and certain kinds may refuse to participate for various reasons. This results in participation bias or volunteer bias. There are four basic kinds of probability samples: simple random samples, stratified random samples, cluster samples, and systemic samples. Simple random samples. This is the simplest kind of probability sample. A sample drawn in such a way that every element in the population has an equal probability of being included, such as in the playing card example above. A random sample is defined by the method of drawing the sample, not by the outcome. If four hearts were picked in succession out of a pack of cards, this does not in itself mean that the sample is not random, although the sample would clearly not be representative. If the sampling method frequently tended to produce four hearts, it would clearly be biased and not truly random. Stratified random samples. In a stratified random sample, the population is first divided into internally relatively homogeneous groups, or strata, from which random samples are then drawn. This stratification results in greater representativeness. For example, instead of drawing one sample of 1,000 people from a total population consisting of 1 million men and 1 million women, one random sample of 500 could be taken from each gender or stratum separately, thus guaranteeing the gender representativeness of the resulting overall sample of 1,000 and eliminating selection bias as far as the gender of the participants go. If 500 women could not be found to participate in the study, but only say 250, it would be possible to correct for the lack of women by multiplying or weighing data obtained from the women in the sample by 2. Cluster samples. Cluster samples may be used when it is too expensive or laborious to draw a simple random or stratified random sample. For example, in a survey of 100 US medical students, an investigator might start by selecting a random set of groups or clusters, such as a random set of 10 US medical schools, and then interviewing 10 students in each of those 10 schools. This method is much more economical and practical than trying to take a random sample of 100 directly from the population of all US medical schools. Systemic samples. These involve selecting elements in a systemic way, such as every fifth patient admitted to a hospital or every third baby born in a given area. This type of sampling usually provides the equivalent of a simple random sample without actually using randomization. Experimental studies. The relationship between independent and dependent variables can be investigated in two ways. Experimental studies in which the researcher exercises control over the independent variables by deliberately manipulating them. Experimental studies are sometimes called intervention studies. Non-experimental studies in which nature is simply allowed to take its course. Non-experimental studies are also called observational studies. In an experimental investigation of a drug's effectiveness, for example, the investigator would intervene, giving the drug to one group of patients, but not to another group. In a non experimental investigation, the researcher would simply observe different patients who had or had not taken the drug in the normal course of events. The hallmark of the experimental method is therefore manipulation or intervention. Experimental studies of new treatments follow a series of stages. In the United States, these stages are regulated by the U.S. Food and Drug Administration, the FDA. Preclinical animal studies are reviewed by the FDA before an investigational new drug application is approved, and clinical trials can begin. Clinical trials fall into three initial phases. Phase 1 studies generally involve a small number, under 100, of healthy volunteers, and the dependent variables are typically the safety, side effects, and metabolic and excretory pathways of the drug. If Phase 1 studies show no unacceptable toxicity, Phase 2 studies are done on somewhat larger numbers of people who actually have the condition being treated. These studies usually have a control group, and the outcomes studied are the clinical effects on the condition and the treatment's short-term side effects and safety. If the drug seems to be effective, phase 3 studies are done. Control trials with larger numbers of patients, typically several hundred or a few thousand, at a range of geographical sites, multi center studies, focusing on efficacy and safety. They often use a range of doses of the drug in different populations of patients. Depending on the findings of phase 3 trials, the developer of the drug may submit a new drug application. The FDA then reviews all the data, confirms that the facilities in which the drug is going to be manufactured meet certain standards, and may then approve or deny the application, request further studies, or request the opinion of an independent advisory committee. Clinical trials. The experimental method in medical research commonly takes the form of the clinical trial, typically a randomized controlled clinical trial, which attempts to evaluate the effects of a treatment. Clinical trials are the gold standard of evidence for cause and effect relationships. However, they have disadvantages. They may pose multiple ethical problems. They are impractical if the cause and effect relationship is one that takes a long time to appear. They are very expensive and time consuming. They require that a treatment is adequately standardized or developed. For example, treatments such as diets, exercise, or psychotherapy may be hard to fully standardize, making the results hard to interpret. Control groups. Controlled clinical trials involve dividing participants or patients into two groups. First, there is the experimental group, which is given the treatment under investigation. And the second group is the control group, which is treated in exactly the same way as the experimental group, except that it is not given the treatment. Any difference that appears between the two groups at the end of the study can be attributed to the treatment under investigation. Control groups therefore help to eliminate alternative explanations for a study's results. For example, if a drug eliminates all symptoms of an illness in a group of patients in 1 month, it may be that the symptoms would have disappeared spontaneously over this time, even if the drug had not been used. But if a similar control group of patients who did not receive the drug experienced no improvement in their symptoms, this alternative explanation is untenable. There are two main types of control groups used in medical research: a no treatment control group, which receives no treatment at all. This leaves open the possibility that patients whose symptoms are relieved by the drug were not responding to the specific pharmacologic properties of the drug, but to the non-specific placebo effect that is part of any treatment. Two, there's a placebo control group, which is given an inert placebo treatment. This allows us to eliminate the explanation that patients in the treatment group were responding to the placebo component of the treatment. The effectiveness of the drug would therefore have to be attributed to its pharmacological properties. In studies of this kind, it is obviously important that patients do not know whether they are receiving the real drug or the placebo. If patients taking the placebo knew that they were not receiving the real drug, the placebo effect would probably be greatly reduced or eliminated. Likewise, the researchers administering the drug and assessing the patient's outcomes must not know which patients are taking the drug and which are taking the placebo. Knowledge or deduction of which groups the patient is in by either the patient or the researcher may cause conscious or unconscious bias. This is assertment bias or detection bias. Specifically, ascertainment bias on the part of the researchers is called assessor bias, and on the part of the patients is called response bias. The patients and all those involved in the conduct of the experiment should therefore be blind as to which patients are in which group. Such studies are therefore called double blind studies. However, it is not always possible to perform a double blind study. For example, in an experiment comparing a drug versus a surgical procedure, it is hard to keep patients blind as to which treatment they received. Although studies have been done in which control group patients have undergone sham surgery under general anesthesia. However, it would be simple for the outcome to be measured by a blind rater who might perform laboratory tests or interviews with the patient without knowing to which group the patient belonged. Such a study would be called single blind study. Under some circumstances, truly controlled experiments may not be possible. In research on the effectiveness of psychotherapy, for example, patients who are placed in a no treatment control group may well receive help from friends, family, clergy, self help books, and so on, and would therefore not constitute a true no treatment control. In this case, the study would be called a partially controlled clinical trial. Clinical trials aim to isolate one factor, such as use of a drug, and examine its contribution to patients' health by holding all other factors as constant as possible. Factors other than the experimental treatment that are not constant across the control and experimental groups are called confounders. For example, in a controlled trial of a new drug versus a placebo for the prevention of prostate cancer, a disease that is known to affect black men more than white men, it would be absurd to allocate all the white patients to the drug group and all the black patients to the placebo group. This would result in allocation bias, and any difference in outcome between the two groups could be attributed to differences between the races rather than to the drug itself. In this situation, patient race is a potential confounding variable. It is likely to contribute differently to the two groups, so that the drug and the placebo groups are not directly comparable with each other. Thus, it is impossible to attribute differences in outcome to the treatment. Note that any effect of the race in this would be an artifact of study design. If race did actually have a true biological effect as an effect modifier, as discussed in Chapter 3, this poorly designed study would not be able to detect it. Confounding is prevented or minimized by several techniques of study design, including randomization, matching, stratification, and restriction. Randomization. Randomization means that patients are randomly assigned to different groups, i.e., to the experimental and control groups. Most clinical trials involve randomization and are therefore called randomized clinical trials or randomized controlled clinical trials. True randomization means that the groups should be similar with respect to race, gender, disease severity, age, occupation, and any other variable, including variables that we do not even recognize, that may affect the response to the experimental intervention. Matching. Randomization cannot guarantee that the experimental and control groups are similar in all important ways. An alternative way of ensuring similarity is by matching. Each patient in the experimental group is paired with a patient in the control group who is a close match on all relevant characteristics. So, if gender, race, age, and smoking status were important factors influencing the course of the disease being studied, each experimental patient would be matched with a controlled patient of the same gender, race, age, and smoking status. Thus, any resulting differences between the two groups could not be attributed to differences in these factors. However, it may be difficult or expensive to find a matching participant for each person in the trial, and variables that are not matched may still turn out to be confounders. Stratified randomization, also called pre-stratification or blocking. This is a combination of randomization and matching techniques. The population under study is first divided or stratified into subgroups, blocks, that are internally homogeneous with respect to the important potential confounding factors, for example, gender, race, age, disease severity. Equal numbers of patients from each subgroup are then randomly allocated to the experimental and control groups. The two groups are therefore similar, but their exact membership is still a result of randomization. Restriction. This involves restricting a study to participants with certain characteristics. If a study is limited to people of just one gender, race, age group, geographical area, and disease severity, etc., then all these factors are removed as potential confounders. Recruitment and retention diagrams show the flow of participants through a study. Figure 5.1 shows an example in the form of a standard for such diagrams from the Consort group. That's the Consolidated Standards of Reporting Trials group, endorsed by hundreds of leading medical journalists or journals, sorry. As figure 5.1 shows, not all participants in a study will produce a complete set of analyzable data. Some participants may not have fully received the treatment they were allocated to. Some may discontinue it, and some may just be missing or lost to follow up. If these participants were equally distributed between the control and treatment groups in a clinical trial, or were similar to the remaining participants in non experimental trials, the only problem this might cause would be a loss of sample size with consequent loss of statistical power. But lost participants are obviously likely to be different. They may have died from the disease under study or been institutionalized, perhaps because of or in spite of the study treatment or the placebo, and died of comorbid illnesses. They may have dropped out due to side effects from or dissatisfaction with the study treatment. They may have had social or transportation problems that made it too hard for them to follow up, and so on. Patients who are lost to follow up are likely to have worse outcomes than those who remain, thus biasing the results of the study in a favorable direction. One way of dealing with the problem of participants for whom a complete set of data cannot be obtained is by intention to treat analysis, in which the data from all participants is analyzed according to the group they were allocated to, regardless of whatever happened subsequently. In other words, it is analyzed according to how it was intended to treat them. This overcomes the problem of bias and resembles the real clinical world in which patients do not take the medications they are prescribed, but it does not solve the problem of participants whose data is missing. This missing data are sometimes filled in or imputed, typically by the last observation carried forward method, in which the last measurement obtained for a given participant is simply carried forward as if it were the same at later dates. For example, a patient who dropped out after 6 months of a 2 year trial of an Alzheimer's disease drug might have his mental status exam scores at 6 months carried forward to the 2 year conclusion. However, this may introduce bias in ways that are hard to disentangle. The patient's last observation carried forward data might make the drug appear to be less effective than it really is if he has failed to get the 2 year benefit of an effective drug, or it may make the drug appear more effective than it really is if the other patients on the drug showed a progressive cognitive decline as the study went on. Hmm. I see. So we can quickly go over this. A template for recruitment and retention diagram. So you enroll, you're assessed for if you're eligible for it. If you're excluded, meaning you don't really meet the restriction inclusion criteria, you decline to participate in or some other reason. But if you are eligible, then you enter a randomized trial, and then you're allocated to a certain group. So you could be allocated to an intervention, received allocated intervention, or did not receive allocated intervention. So you're given two um, kinds of intervention, and then you have to follow up that patient. Now you could lose that person, or For some reason, the intervention was discontinued, and finally, um, analyze excluded. Then you analyze it and see if it was if why it was not analyzed. Hmm. Okay. If a study is designed and performed well, including in its management of missing data, so that it is free of bias and alternative explanation for its findings are ruled out, it has internal validity. It will be valid for the particular participants it studied. If the sample is truly representative of the population from which it was drawn, then the study has external validity. Its findings can be applied to that population. However, it may not necessarily be generalizable, valid more broadly to other people in other places or at other times. All clinical trials will be limited to some extent, if only by the geographical area of the participants, such as an area within reach of the medical centers running the trial. And in most clinical research, participants will also be limited by certain entry or inclusion criteria, which must be clearly specified before the sample is drawn. For example, in a trial of a new drug for Alzheimer's disease, there may be criteria specifying patients' ages, how the diagnosis was made, how long they have had the disease, and how severe it is, the absence of certain comorbidities, and that they have not already taken any drug for the disease. The findings of such a study may not be generalizable to the real world Alzheimer patient, who are likely to be much more varied, complex, and less well defined. Entry and exclusion criteria have an ethical dimension if the sample studied is not representative representative of the population of people with the disease in question, or of the population as a whole. Until the 1980s, studies showed a notorious tendency to be restricted in favor of males and whites. But it is now a matter of public policy in the United States that proposed studies cannot restrict studies in these ways without strong justification. Research ethics and safety. All research has an ethical dimension, and in the United States, researchers are required to obtain permission from an institutional review board before collecting any human data and must inform the institutional review board of any changes as the study progresses. The membership and procedures of institutional review boards are federally regulated. An institutional review board may be part of an academic institution, a hospital, or it may be a freestanding for profit commercial entity. Institutional Review Board rules grew out of the scandal of the Tuskegee syphilis experiment, in which poor black men in rural Alabama who had been infected with syphilis were studied for several decades without being treated, some for many years even after penicillin was established as a standard and effective treatment in 1951. Institutional Review Board rules require that participants' autonomy is respected. It requires that vulnerable people are protected, such as the elderly, seriously ill, and prisoners, and that those with diminished autonomy, such as children and the mentally ill, are also protected. The rules also require that benefits are maximized and potential harms minimized. Further, they require that the consent to participate is truly informed consent. The potential participant must be given sufficient information in a way that he or she can understand, taking into account limitations of intelligence, rationality, maturity, language, etc. And the consent must be voluntary, not the result of coercion or of excessive or inappropriate reward. Clearly, the amount of information given will vary with the type of study being done. The risk of a trial the risk of a trial of new type of prosthetic heart valve is greater than the risk of just having blood drawn. Just as with consent to medical treatment, the informed consent form that participants sign is not the consent itself, but is only a record of the consent. It does not replace the process of obtaining informed consent. The institutional review boards are also required to ensure that standards are in place to ensure the privacy of research participants and maintain the confidentiality of data about them. Institutional review boards and the medical community as a whole are paying increasing attention to financial and academic conflicts of interest among researchers and their sponsors, which potentially cause many unethical biases in the design, recruitment, conduct, interpretation, and reporting phases of clinical trials. Ethical problems are particularly significant in experimental studies, especially if there is reason to believe that the independent variable under investigation is either a harmful one or a strongly beneficial one, even if these reasons only become apparent during the course of the study. For example, although it would be unethical to test the hypothesis that it takes 15 years of heavy alcohol drinking to cause cirrhosis of the liver by conducting an experiment, this hypothesis can be tested observationally by finding people who have done this in the ordinary course of events. Similarly, if it is believed that an effective treatment for a condition already exists, the use of a placebo or no treatment control group may be unethical. As a result, some long standing, time honored medical interventions may be hard to study ethically or legally because they have become the standard of care, despite no real studies having been done to prove their efficacy. In the 1950s, experiments on the effects of oxygen on premature babies were opposed on the grounds that the control group would be deprived of a beneficial treatment. Later, when it became strongly suspected that excessive oxygen was a cause of a type of blindness, retinopathy of prematurity, similar experiments were opposed because the experimental group might be subjected to a harmful treatment. One solution is to assign the control group to a different active treatment. The new experimental treatment is then compared only with that treatment. This may be problematic for some research sponsors, such as pharmaceutical companies, who may wish to show that their new drug is more effective than a placebo, rather than subject it to a head to head contest with its competitors, especially as FDA approval rests on the drug being effective only versus a placebo, not on it being more effective than or even as effective as existing treatments. Another solution, if the treatment is not time sensitive or if resources are inadequate to treat all patients at the same time, is to use a waitlist control group. The control group patients will receive the treatment after the experimental group patients have received theirs, so the control's treatment is merely delayed, not denied. Related to, the, Related to this is the crossover study, in which half the patients received the active treatment for a period, followed by the placebo, while the other half received the placebo first, followed by the experimental treatment. This kind of study is called a within subjects design. Comparisons between the drug and placebo are made within each subject or patient rather than between different subjects. In these designs, each patient serves as his or her own control. So the control group is the same subject control group. This is also called a repeated measured study because the measurements of the dependent variables, such as symptom severity, are repeated within each patient rather than a single measurement being taken and compared between different patients in the more common between subjects designs. If there is a danger of a carryover effect, for example, if the treatment is a drug that may continue to have some effect after it is withdrawn, there can be a washout period between the drug and the placebo phases, during which no treatment is given. Figure 5.2 illustrates a crossover design with the washout. One group of patients receives the drug for 1 month and then crosses over to receive the placebo after a 1 month washout. The other group follows this pattern in reverse order. So here's the picture of a crossover design with washout, where you have two groups, and the first month, group A gets the drug and the other one gets the placebo. Second month, um, you have the washout period, and then you just flip the groups over. Group B will get the drug, and Group A will get the placebo. The placebo. Hmm. As more treatments become available for many diseases, and as it becomes harder to create truly novel treatments, non inferiority trials are becoming common. This kind of clinical trial seeks to show only that a new treatment is not inferior to an existing one. This may be done for commercial reasons or because the existing treatment may have significant problems, such as side effects requiring lab monitoring, having to be given by injection or multiple times a day, etc. In non inferiority trials, the null hypothesis is that the old treatment is more effective than the new one, and the alternative hypothesis is a directional one, that the new treatment is at least as effective as the old one. This is therefore a situation in which one tail statistical testing is appropriate, as discussed in Chapter 3, because there will be no practical difference between finding a difference that favors the new treatment and finding that there is no difference between the two treatments. In either case, the new treatment is simply at least as effective as the old one. Major drug trials are subject to regular safety monitoring to check that participants are not being subjected to unexpected or excessive risks, and that certain points at interim analysis will be performed, for example, after 1 year of 3 year trial, or after 50% of participants have been enrolled for several reasons, which are to check for risks for the treated patients, so called safety monitoring, to see whether the treatment has such significant benefits that it would be unethical or unnecessary to continue the study, depriving the control group patients of the treatment, and to see whether there is such a small chance of the study producing a useful result that it is futile to continue it. Several trials have been discontinued for each of these reasons in recent years after the interim analysis were followed by the application of pre-specified stopping rules or stopping boundaries. There is concern that in some cases, trials may be stopped prematurely before truly robust conclusions can be drawn for purely commercial reasons, such as to allow a new drug to be brought to the market or to gain an approved indication more quickly. A solution to potential ethical problems caused by randomizing participants to different treatments is to conduct a patient preference trial, in which patients who have preferences about their treatments are allocated to that treatment. A patient preference arm can be included in a regular randomized trial. Patients who have a strong preference are given the treatment they want, while those who have no preference are randomized. This allows increased numbers of patients to be studied. For example, if there are two different kinds of drugs for the disease under study, some patients may refuse to participate in the randomized trial, but may be willing to enroll in one in which they get the medication they prefer, and has the advantage of reflecting real clinical practice, where, where patients do express preferences about their treatments and are more likely to be motivated to comply when they are given the treatment they want. Non experimental studies. Non experimental or observational studies fall into two general classes: descriptive studies and analytical studies. Descriptive studies aim to describe the occurrence and distribution of disease or other phenomena. They do not try to offer explanation or test a theory or a hypothesis, but merely attempt to generate a description of the frequency of the disease or other phenomenon of interest according to the places, times, and people involved. These studies will use descriptive statistics, but not inferential statistics. Descriptive statistics Descriptive statistics I'm sorry, descriptive studies are often the first method used to study a particular disease. Hence, they are also called explanatory studies. They may serve to generate hypotheses for analytical studies to test. There are many well known examples. The first finding that occupational environmental exposures could cause cancer. In 1775, Sir Percival Pott, a British surgeon, described scrotal cancer commonly found among boys who were employed as chimney sweeps. Before this, cancer was assumed to be the result of human biology only. A description of 8 unusual cases of Kaposi sarcoma among gay males in California, which was earliest study of AIDS. As with scrotal cancer, the true cause of the disease was unknown at the time, but the description generated useful hypotheses. Analytical Analytic studies aim to test hypotheses or to provide explanations about a disease or other phenomenon. Hypotheses or explanations that are often drawn from earlier descriptive studies. They therefore use inferential statistics to test hypotheses. Descriptive and analytic studies are not always entirely distinguishable. For example, a large scale descriptive study may provide such clear data that it provides an answer to a question or gives clear support to a particular hypothesis. Non experimental designs. Descriptive or analytic studies use one of four principal research designs. They may be cohort studies, case control studies, case series studies, or prevalence surveys. Cohort studies. Cohort studies focus on factors related to the development of a disease. A cohort, a group of people that does not have the disease of interest, is selected and then observed for an extended period. Some members of the cohort will already have been exposed to a suspected risk factor for the disease, and others will eventually become exposed. By following them all, the relationship between the risk factors and the eventual outcomes can be seen. This kind of study, therefore, allows the incidence and natural history of a disease to be studied. Some cohort studies, however, do follow patients who do have the disease of interest. In particular, inception cohorts consists of patients who have just been diagnosed and who are followed to determine their prognosis. Determining which people are classified as members of the exposed versus unexposed groups involves a decision called exposure allocation. For example, if we are interested in examining the relationship between heavy alcohol use and cirrhosis of the liver, we would need to define what constitutes heavy alcohol use and what does not. Cohort studies may be loosely termed follow up or longitudinal studies because they follow people over a prolonged period, tracing any changes through repeated observation. They are also called prospective studies because people are followed forward from a particular point in time. So the researcher is prospecting or looking for data about events that are yet to happen. Cohort studies are sometimes also called incident studies because they look for the incidence of new cases of the disease over time. A famous example of a cohort study is the Framingham study, which started in Framingham, Massachusetts in 1949, with a cohort of more than 5,000 people who were free of coronary heart disease. The individuals in the cohort were re examined every 2 years for more than 30 years, and it allowed the first identification of the major physical risk factors for coronary heart disease. Cohort studies have a number of significant advantages. The take home point here is that when a true experiment cannot be conducted, whether for ethical or practical reasons, cohort studies are the best form of investigation. Their findings are often extremely valuable. Also, the cohort studies are the only method that can establish an absolute risk of contracting a disease, and they help to answer one of the most clinically relevant questions. If someone is exposed to a certain suspected risk factor, is that person more likely to contract the disease? Cohort studies may also reveal the existence of protective factors such as exercise and diet. Because cohort studies are prospective, the assessment of risk factors in these studies is unbiased by the outcome. If the Framingham study were retrospective, for example, people's recollection of their diet and smoking habits could have been biased by the fact that they already have coronary heart disease. This effect is known as recall bias. In addition, the chronological relationship between the risk factors and the disease is clear in prospective studies. For the individuals in the cohort who ultimately contract the disease of interest, data concerning their exposure to suspected risk factors has already been collected. However, in a retrospective study, this may not be possible. Again, if the Framingham study were retrospective, it might have been impossible to obtain accurate information about the diet and smoking habits of people who had already died. Other types of studies are often unable to include people who die of the disease in question, often the most important people to study. Information about suspected risk factors collected in cohort studies can be used to examine the relationship between these risk factors and many diseases. Therefore, a study designed as an analytic investigation of one disease may simultaneously serve as a valuable descriptive study of several other diseases. They allow the chronological relationship between exposure to the risk factor and development of disease to be determined. Cohort studies have some important disadvantages as well. For instance, cohort studies are very time consuming, they're laborious, and expensive to conduct. Members of the cohort must be followed for a long time, often for many years, before a sufficient number of them get the disease of interest. It may be very expensive and difficult to keep track of a large number of people for several years, and it may be many years before results are produced, especially in the case of diseases that take a long time to appear after exposure to a risk factor. Another disadvantage of cohort studies is that they may be impractical for rare diseases. If one case of a disease occurs in every 10,000 people, then 100,000 people will have to be followed for 10 cases to eventually appear. However, if a particular cohort, such as an occupational cohort, exists with a high rate of the disease, a disease that is rare in the general population can still be studied by this method. A classic example was a study of workers in a vinyl chloride factory and their subsequent development of angiosarcoma of the liver. Historical cohort studies. Although cohort studies are traditionally prospective, there is second kind of cohort study, the historical or retrospective cohort study, in which the researcher has access to information about the prior exposures of a particular group of people and looks back at the records of all the people in this group to see whether exposure is related to disease. A memorable, if trivial, example is the study of the mummy's curse, the notion that people who were associated with events surrounding the opening of King Tutankhamun's tomb in Egypt in the 1920s would die prematurely. Done in Nelson 2002. The author retrospectively reviewed data about the survival of two cohorts of Westerners. One cohort was simply present in Egypt at that time, while the other cohort was present with the archaeologist Howard Carter, either when either when tomb, the sarcophagus, or the coffin were opened, or when the mummy was examined. No association was found between this exposure and premature death. Case control studies. While cohort studies examine people who are initially free of the disease of interest, case control studies compare people who do have the disease, the cases, with otherwise similar people who do not have the disease, the controls. Case control studies start with the outcome or dependent variable, the presence or absence of the disease. They then look back into the past for possible independent variables that may have caused the disease. to see whether a possible risk factor was present more frequently in cases than in controls. Hence, they are also called retrospective studies. One classic exploratory case control studies uncovered the relationship between maternal exposure to diethylstilbestrol (DES) and carcinoma of the vagina in young women, done by Herbst in 1971 and his group. 8 patients with this rare cancer were each compared with 4 matched cancer free controls. Looking back at their individual and maternal histories, no significant differences appeared between the cases and the controls on a wide range of variables, but it was found that mothers of 7 of the 8 cases had taken diethylstilbestrol in early pregnancy, 15 to 20 years earlier, while none of the 32 controls mother had done so. Case control studies are like historical cohort studies in that they both look back at the data, so they are post hoc studies. But they differ in that all the people initially identified in case control studies have the disease. Case control studies offer some significant advantages. Here's an important point. They can be performed fairly quickly and cheaply, especially in comparison with cohort studies, even for rare diseases or diseases that take a long time to appear, as the vaginal carcinoma example shows. Because of this, case control studies are the most important way of investigating rare diseases and are typically used in the early investigation of a disease. Case control studies require comparatively few subjects, and they also allow multiple potential causes of a disease to be investigated. Case control studies also have a number of disadvantages and are particularly subject to bias. For instance, people's recall of their past behaviors or risk factor exposure may be biased by the fact that they now have the disease. This would be a recall bias. The only cases that can be investigated are those that have been identified and diagnosed. Undiagnosed or asymptomatic cases are missed. People who have already died of the disease cannot be questioned about their past behaviors and exposure to risk factors. Selecting a comparable control group is a difficult task that relies entirely on the researcher's judgment. Also, they cannot determine the rate or the risk of the disease in exposed and non exposed people. And they cannot prove a cause and effect relationship. Case series studies. A case series simply describes the presentation of a disease in a number of patients. It does not follow the patients for a period, and it uses no control or comparison group. Therefore, it cannot establish a cause and effect relationship, and its validity is entirely a matter for the reader to decide. A report that has 8 of a series of 10 patients with a certain disease have a history of exposure to a particular risk factor may be judged to be extremely useful or almost worthless. Despite these serious shortcomings, series studies are commonly used to present new information about patients with rare diseases, and they may stimulate new hypotheses. Again, the original work on AIDS patients described just 8 cases of Kaposi sarcoma. Case series studies can be done by almost any clinician who carefully observes and records patient data. A case report is a special form of case series in which only one patient is described. This too may be very valuable or virtually worthless. Prevalence surveys. A prevalence survey or community survey is a survey of a whole population. It assesses the proportion of people with a certain disease. The prevalence of the disease to be discussed in the next chapter. So prevalence surveys assess the proportion of people with a certain disease and examines the relationship between the disease and other characteristics of the population. It may also be used to find the prevalence of a disease in people who have and have not been exposed to a risk factor. For example, chronic obstructive pulmonary disease in smokers versus non smokers. This is the prevalence ratio. Because prevalence surveys are based on single examination of the population at a particular point in time and do not follow the population over time, they are also called cross sectional studies, in distinction to longitudinal cohort studies. Prevalence surveys are common in the medical literature. For example, a study could compare the prevalence of coronary heart disease in one community versus a different community with different dietary or exercise habits. Another example would be a study examining the prevalence of lung disease in one city, which could then be compared with another city with lower levels of cigarette consumption or air pollution. Prevalence surveys suffer from a number of disadvantages, such as because these prevalence surveys look at existing cases of a disease and not at the occurrence of new cases, they are likely to overrepresent chronic diseases and underrepresent acute diseases. Prevalence surveys may be unusable for acute diseases, which few people happen to have at the moment they were surveyed. Also, people with some types of disease may leave the community or may be institutionalized, causing them to be excluded from the survey. Findings of prevalence surveys must be interpreted cautiously. The mere fact that two variables such as high fish intake and low prevalence of coronary disease are associated does not mean that they're causally related. Although there are expensive and laborious, prevalence surveys are common because they can produce valuable data about the wide range of diseases, behaviors, and characteristics. These data can be used to generate hypotheses for more analytic studies to examine. Figure 5.3 summarizes the chronological sequence of events in the major kinds of non-experimental study designs. So here we have a cohort study um, where you have some person who was exposed, you start studying the charts, and they're further exposed to some things, and maybe they have disease or not have disease, don't know. Historical cohort studies, so basically you start the study first, and then you go back and see, oh, this is a disease, and was Y responsible for that disease? Um, and then you have these case control studies where the study will start and you would look back at the diseases and you will look at, okay, so these people had the disease and this is the cause, this seems like the cause of the disease. So let's follow this through now and see if this exposure is going to cause disease in more people. And then you have case series studies where you start with the study and then you say, okay, this is a disease and definitely this is exposure, I believe. And finally, you have prevalence surveys where you just walk around and you just say, okay, so you have the disease and this is what the exposure was. Okay. Ecological studies. An ecological study is any kind of study in which data is collected about a whole population, a large group or community, and is analyzed at that level. Examples would include studies of the rate of diabetes in countries with different levels of automobile ownership, studies of homicide rates in states with different firearm laws, and studies of lung cancer rates in places with different tobacco taxes. They can usually be done quickly and inexpensively using existing bodies of data. Unlike cohort studies, no data is available or analyzed about individuals. For example, an ecological study may show that communities with large numbers of fast food outlets have higher rates of diabetes, but it would not gather data about whether an individual's food choices relate to his or her risk for diabetes. Ecological studies need not be non-experimental. Community intervention trials are experimental trials done on a population or community wide basis. The experimental group consists of an entire community, while the control group is an otherwise similar community that is not subject to any kind of intervention. An example is the COMMIT trial, in which the rates of quitting smoking were compared in 11 pairs of North American communities. The experimental communities in which multiple smoking education activities were conducted, such as media campaigns, and the match control communities in which no such intervention was performed. This was the Fisher study in 1995. Post marketing studies. A special kind of non experimental study is post marketing surveillance, also known as Phase 4 or pharmacovigilance studies. These involve gathering data about patients taking a particular medication that is already in general use. They may be used to help evaluate the treatment's effectiveness, but in particular, they monitor its safety. An adverse effect that occurs in, say, 1 in 5,000 patients, or just in particular kinds of patients, may remain undetected in a clinical trial, but it will become apparent if hundreds of thousands or millions of people are followed in phase 4 studies, especially as these people will have multiple comorbidities and take multiple other medications. Such studies may be required by the FDA or may be done by the drug manufacturer for marketing or other purposes. In the United States, these studies are conducted both actively and passively. Active post marketing studies are done by the FDA's Center for Drug Evaluation and Research, CDER, Office of Surveillance and Epidemiology, using reviews of hospital or outpatient medical records, pharmacy databases, and laboratory data, or by establishing registries, example of all pregnant women who have been prescribed a certain drug. Passive post marketing studies is done by the FDA MedWatch program, in which doctors and the general public voluntarily report adverse reactions. These processes may show long term negative outcomes, rare adverse effects, or even simple problems such as confusion between drugs with similar names. And post marketing studies has led to several drugs being withdrawn from the market, having warnings attached, being limited to certain kinds of patients, or simply being renamed.